Good morning, everyone. My name is Sona. I am on the Junior Advisory Board. I am also an osteo warrior. I'm about 5 years out of treatment. And I just graduated from Tufts and I'm starting at Harvard Medical School in August. Thanks. And now I have the privilege of introducing Dr. Reed. So Dr. Reed is a pediatric hematologist oncologist on the MSK Kids team at Memorial Sloan Kettering Cancer Center. He specializes in the care of children, adolescents, and young adults with sarcomas and other solid tumors. His clinical and research focus centers on understanding the changes that occur in tumors that lead to resistance to therapies. Recognizing that cancer is complex and evolves over time, Dr. Reed and his colleagues aim to uncover why this resistance develops in order to better predict tumor behavior and design therapies that address resistance and reduce relapse. Their ultimate goal is to restore tumor sensitivity to anti-cancer treatments and help young people with cancer live longer, healthier lives. And with that, I'll pass it on to Dr. Reed.
Thank you very much. I'm a substitution for Dr. Gatenby, who's ill, so couldn't travel. He was supported by the Because of Charlotte Travel Award, and I believe Charlotte's father, Sherrick, is in the audience. A quick thing about Charlotte was a senior in high school when she became an osteo angel and started her battle against osteosarcoma when she was 15. I didn't know her, but this biography is fantastic, and during 3+ years, she demonstrated remarkable courage and strength, athletic, funny, stubborn, creative, full of grit, and a sweet friend to all. She loved her family, friends, fashion, cooking, sports, the beach, anything cozy, and her dog, Cody, and a sewing business called That's So Char, S-E-W, which sounds fantastic. It was started donating funds to osteosarcoma research at UCSF and MIB agents. She was on the junior board position with MIB and became an angel in 2021. So thank you for that support. Bob Gatenby, just so you know, had a huge influence on my life. Bob, and you've heard these themes, you keep kind of hearing them. I'm going to focus on the we and the we persevere. You heard about all this data we don't use, and Bob Gatenby was a physicist at Princeton and then became a radiologist, and when he looked at his first scan, he thought, man, this is great, and then he talked to the oncologist, and the oncologist said, how big is the tumor? Like, is it 8 centimeters or 8.2 centimeters? And he said, there are thousands of pixels, and there are thousands of images. Like, there's a million data points here. Like, you only want to know how big the tumor is? And the oncologist was like, don't be weird. Just, I need to know how big the tumor is. Read the next scan. I need to read. And so Bob said, no, like, this is not right, and it's not linear, and my God, like the weather, how about the weather? Like, weather's complex. Can I convince oncologists that cancer's like the weather? It's a complex system. No, he couldn't. And so he hates dogma, and he felt like the dogma of oncology was totally wrong. And so he and this other Bob, the Bobs at Moffitt, Bob Gillies and Bob Gatenby, just rallied against everything. And so they would love to start a conversation with saying cancer and DNA have nothing to do with each other. And people would get all up in arms, like, we're going to sequence tumors, we're going to find all the things, and we're going to figure it all out. But Bob would go to these math camps, retreats, conferences like this, and he'd be the only clinician. And the math people would make fun of him. Math people making fun of you is a pretty big low. And so, but Bob would do it. He did it over and over and over again. And the math people said, this guy actually knows what he's doing. He's pretty good at math. And he told a guy, Sandy Alexander, hey, I'm going to be this great radiologist. I'm going to be the chair of a department someday, and I'm going to recruit you, and we're going to make the first integrated mathematical oncology program in the country. And Sandy said, whatever. And he went back to Scotland. And then Bob left Arizona, came to Moffitt in Tampa, and recruited Andy to become the chair of integrated mathematical oncology. And math models and ideas of different ways of approaching cancer started to take root. Now, being different, being oppositional is not super easy. Challenging dogma is not super easy. And so Bob, he did a lot of different things, but the cancer that he approached was prostate cancer, and the approach that he had was using good drugs intermittently rather than continuously using them. And so he knew that if you had a drug that induced a response but invariably led to resistance and death, that a better way to do that would be to give it intermittently. He used lessons from agriculture. Farmers do not spray pesticides to try to get rid of every bug on the planet. They actually just try to keep the bugs down to a low level, and they start and stop, and they start and stop. And so he showed that you can give half of a drug, abiraterone, to prostate cancer patients, and they'd live 3 times longer. But it wasn't a randomized trial. Well, it didn't do a lot of things, so it wasn't perfect. But when I was trying to do targeted therapy, and I did not have a ton of success with osteo and Ewing, when I tried to do immunotherapy and with the quick things, the checkpoint inhibitors, didn't see a strong signal. I was kind of a little bit lost at Moffitt in 2018, and Bob was also kind of in the penalty box for having ticked off a couple administrators, which was common. And so we were a forced marriage, and people said, write a trial like Bob in pediatrics. And so Bob said, you don't do very well in pediatric oncology. You guys a lot of times fail. You should do what we did in prostate cancer. And I said, yeah, I guess so. Mathematically, you might be right. But Bob, tell me how you would tell a parent of a newly diagnosed patient we should start and stop chemo and give up on a cure. And he said, you got me. That's not easy. So he and I started working on, he said, we need to make the tumors go extinct. And I said, yeah, that's a weird way of thinking about it. Patients want a cure. And he's like, well, it's really extinction, so we need to learn about extinction biology. And so Bob went down, as he always does, this crazy wormhole, super smart. And he said, do you realize 99% of things on the planet went extinct? I was like, no. He says, most of the time, it's not like an asteroid. It's this series of small events. And I was like, oh, that's interesting. We should probably do a series of different chemos. And I was like, that's exactly what we do for pediatric leukemia. And so we started this extinction therapy kind of idea. So it would have been great if Bob was here doing this himself, but I do want to recognize the giant. I mean, he's in a museum in London because of his ideas. He's a really good person, and we'll get him back here some other time. But he's perfect for introducing, and I am his protege in many ways in trying to design the best studies to do that, to hit cancer over and over again, learn what changed, and kind of keep eradicating it. So with that, I'll introduce the speakers and the topic of mechanisms of resistance and evolution. So really excited about this. I think that we don't always need new, fancy, shiny drugs. I think there's lots of drugs out there that work in cancer that we need to repurpose. And so some of the themes that you'll hear today are drugs that are already out there, already FDA approved, that have some activity in osteosarcoma. I think, newsflash, probably they're not the magic cure for things, but probably they do something. And so part of a strategy that we could do towards better treating osteosarcoma. And now let me introduce the speakers before I become the first flag-shamed moderator. So you'll first hear from Dr. Scotlandi, an interesting project finding something about ixabepilone that I'm looking forward to learning about. Dr. Mahajan from my small hometown of New York City will be presenting as well, along with Dr. Burke, who trained with us at MSK and is now doing excellent things at Stanford. Dr. Lizardo is going to bring his skills from NCI and the Sorensen lab and talk to us about his work. And then Sasha Blay will also be talking about her work at the Hospital for Sick Children. So I'm looking forward to all these presentations. And without further ado, Dr. Scotlandi.
So good morning to everybody. I'd like to thank you all for giving me the opportunity of being here and to present our recent work in osteosarcoma. I don't see the slide, so okay. So I work in Italy, and I work at the Rizzoli Instituto, which is an hospital that is devoted to study and cure bone disease, including bone sarcomas. So just a few words of introduction, just to say that osteosarcoma, as we all know, is a disease that is characterized by a strong genomic heterogeneity. But the heterogeneity is not only at the genetic level, is also at biological level, because we know that the transformation event can occur in cells at a different stage of their differentiation process. So we can have a different type of tumors depending on the stage of the cells that is transformed. And heterogeneity is also at an epidemiological level, because Even if osteosarcoma is a disease of children and young adults, we have a small peak of incidence in the elderly, and the causes events is probably very different in one type of osteosarcoma and the other. So all that just to say that modeling osteosarcoma is really very difficult, and likely we don't have a perfect model, and probably we will never have a model that can be representative of all these general level of heterogeneity. So I think that the key is to choose the right model for the right purpose and to put the information in the appropriate cellular context. Yesterday, you heard that there were quite a lot of models around. Among them, patient-derived xenografts are certainly one of the best tools that can be used to test novel therapeutic approaches. Several groups, several labs around the world have developed PDX, including the lab of Alejandro, who was one of the first. In our institution, we end up with a collection of 30 patient-derived xenografts that are representative not just of tumor from young people, but also from older people. These tumor, we have some rare PDX from rare histological variants, such as the telangiectasic tumor osteosarcoma or the extraskeletal osteosarcoma. We have PDX from primary tumor before and after chemotherapy and from metastatic lesion. We also have tumors that PDX that comes from very aggressive tumor, because in most of the cases, the patients, the tumors that developed the PDX were coming from patients who died of the disease. And in a certain percentage of cases, there is overexpression of P-glycoprotein, which is a mediator of chemoresistance, at least resistant to doxorubicin. And this expression was there from the very beginning. So it means that we are modeling not just acquired resistance, but we are modeling also innate resistance. So, but however, working with okay, so we confirm, like many others, that these PDX maintain the same genetic alteration of the original tumor. So the genetic alteration are more or less always the same. Alteration in the oncosuppressor RB and P53, or alteration in some oncogenes such as MYC. So these are very good models, in my opinion. However, using PDX for drug screening is time and cost consuming, and it can lead to some very heterogeneous results. So as like other group in the world, we have developed a PDX-derived cell line. You can see that it is much easier to obtain cell lines from PDX than from human sample directly. These PDX-derived cell lines maintain the genetic alteration of the original tumor, and we re-inject this cell line in mice to see how aggressive they were. So you can see that there are quite a heterogeneous behavior here. We have some cell lines that grow very well after and develop tumor, but the same cell line do not form a metastasis or vice versa. So we were able to confirm that the tumors, even in this condition, maintain the genomic alteration of the original human tumor, but we were never able to connect this heterogeneity behavior with a specific genetic alteration. So it means that each cell lines, so potentially each patient is still different from each other, even if we found some genomic commonalities. So that's why we decided to choose a different approach. So we select our most aggressive cell line. It is a cell line that has a huge amplification of MYC, 100 copy number gain, and extra chromosomal DNA. And this cell line came from a patient who died in 8 months without any response to any type of treatment. So using this cell line, okay, we perform a drug screen library with a drug library composed of 2,700 compounds, all of them FDA approved. We use two drug concentration of the and we selected the drug that were able to inhibit the growth at a level higher than 55%. Then we combine the results with the two concentration, ending up with 44 compounds, most of them approved for cancer. These 44 compounds can be classified in 7 subtypes, subgroups, according to their mechanism of action. So we select one drug for one of these subtype, and we validate the efficacy of these 7 drugs against all the panel of PDX cell line that we have developed. And we were able to validate 5 out of 7 drugs. And for 2 of these cell drugs, I think we got a very interesting result. So we went attracted to ixabepilone because this is a tubulin stabilizer, and this drug is not a substrate of P-glycoprotein, so it should be active against tumors that express P-glycoprotein from the very beginning. The second drug was omarinctonin. This drug is a general protein translation inhibitor, and it is approved for the treatment of chronic myeloid leukemia. And we got interested in this drug because there is a report indicating that it can inhibit also the expression of MYC. So both of these 2 drugs are very active in vitro at nanomolar concentration. We extend the evaluation to a larger panel of cell line, and the drug was confirmed to be active. Particularly, they were active also in the 2 cell lines that we have developed that are cell lines that are resistant to methotrexate and cisplatin. And you can see that the activity of ixabepilone and omarinctonin is the same as the parental cell line. The activity was also very good when the cell lines were grown in 3D condition. So you can see here that the efficacy of ixabepilone and omarinctonin is maintained still at a nanomolar concentration, and you see that there is a very good inhibition of spheroids there, so in a more different condition. However, when we re-inject the cell line and we treat in mice and we treat xenografts, we observe quite a different behavior, level of efficacy between omarinctonin and ixabepilone. You can see here the growth curve inhibition of ixabepilone. I think that these are very nice. In my opinion, one of the best that I have ever seen in my career, because there is, in some cases, in the right of when the single mice are shown, in the right part of the slide, you can see that we observe some truly regression in the tumor. And this was not the case with omarinctonin, because in that case, we still observe an inhibition of the tumor volume, but we don't have any regression, we just have a stabilization of the tumor. So in our hand, no doubt that ixabepilone has a higher efficacy compared to omaritinib. It's not the first time that ixabepilone is used. There was just this paper 20 years ago from the St. Jude group. They also treated osteosarcoma PDX, and they also found that osteosarcoma is a challenging tumor to be treated, but still they found in some of these osteosarcoma PDX a complete regression. So based on this evidence, preclinical evidence that were more or less the same as ours, there are these two studies, clinical studies. The first one was a phase 1 clinical study with the idea to find, to identify the toxicity, and the paper end up with the idea that the drug has a manageable toxicity. The second one was a phase 2 clinical study, and unfortunately, this study reported negative results. So they didn't observe any response in the childhood tumors that they tested, including 11 osteosarcoma. So of course there are inconsistencies between preclinical and clinical studies. I think this may be due to several factors, including the fact that the patients that were tested in the phase 2 clinical study were very heavily pretreated. The drug and the schedule used in preclinical and clinical conditions were different, and also the clinical trial did not consider stable disease as a primary endpoint. So this is not a good news, of course, but I still think that this drug deserves an opportunity to be studied deeper. I show you that it was very effective against a very aggressive cell line with a huge overexpression of MYC. So we are now wondering if there is a connection between its activity of tubulin stabilizer with the MYC activity in the cell. So that's all, and I'd like to thank, of course, the people at Rizzoli that helped me in this research, the funding, and also the family of Isaac who helped me to being here. Thank you.
Okay, I'd like to thank the organizers and Christina for inviting me to give a talk on this very early stages of this new project I've been working on, which is a follow-up on my previous publication. So I always like to start my talks with this picture that basically describes or displays fluorescent human osteosarcoma cells that just arrived in the lung. So that's the red stain there that you see in the lung. So those are human metastatic osteosarcoma cells, MG63.3, and this is about day 0, about 20 minutes after a tail vein injection. So the main question that I've been trying to grapple with for a while now is how do these metastatic osteosarcoma cells survive in the lung? I'm interested in how they actually adapt to this harsh microenvironment. And if we can understand their adaptation mechanisms, we can develop new strategies to develop anti-metastatic therapeutics. So today I'm going to talk to you about targeting the mitochondrial oxidative stress response in metastatic tumor cells as they try to adapt to oxidative stress. So yesterday, Kelly Guptill described a bottleneck that occurs when the tumor cells arrive in the lung microenvironment. And if you look in the metastasis literature, you see that people have known that when tumor cells arrest in the lung microvasculature, they interact with cells like the microvasculature, the pulmonary microvascular cells, as well as macrophages and such. In particular with microvascular cells, they actually induce the release of a burst of ROS when they arrive in the lung, and that ROS is responsible for clearing most of these melanoma cells that arrive in the lung. And what I was fortunate enough to do my master's in this lab, and what they did was they modified or modulated the mouse's ability to secrete that ROS, and they were able to increase pulmonary metastasis. So that's one major perturbagen that these tumor cells encounter when they arrive in the lung. And over the years, over the past 20 years, people have demonstrated that when tumor cells arrive in the lung, they adapt to that oxidative stress. In melanoma, as well as in breast cancer cells, they adapt this unique oxidative stress response. And more recently, this last fall, my work demonstrated that in several models of metastatic osteosarcoma cells, that through the NRF2, which is a major transcription factor for the antioxidant responses, the translation of that protein is required by EI4A expression, and we found that through a tool compound and a clinically investigated drug called sotatercept was able to inhibit NRF2 translation, and that potently reduced the amount of metastases. So NRF2 is responsible for many pathways for adaptation, and actually about 4 or 5 of those pathways converge on mitochondrial biology. So the green mitochondria that you see there is, you're not meant to memorize that, but just to show where all the red dots are, where NRF2 has an influence on that particular pathway. So how do you choose what to go after as a potential anti-metastatic therapeutic? NRF2 regulates, for example, antioxidant and detoxification enzymes in the mitochondria, mitochondrial biogenesis, mitophagy, and electron transport chains, and so on. So one of these targets called PRDX3 was attractive because in an online data set actually generated by James Morrow, in that data set we found that only when the tumor cells arrive in the lung microenvironment in the PUMA assay do the highly metastatic cells have a differentially upregulated expression of PRDX3. And then other labs have found that PRDX3 through a CRISPR knockdown was one of the genes that were crucial for the adaptation to oxidative stress. So PRDX3 can be inhibited by theostreptin, a natural compound which is used as an antibiotic. And theostreptin forms the active compound of a clinically investigated drug called RSO-021, which I think they completed a phase 1 trial and identified a maximally tolerated dose, and they're going into phase 2 clinical trials. So what is peroxyredoxin? So it's a unique type of antioxidant enzymes that are responsible for neutralizing hydrogen peroxide as well as lipid peroxidases and peroxynitrite into more or less toxic species. There are 6 of them, and from 1 to 6, and each of them have a unique location. 3, 5, and 6, and 1 are regulated by NRF2, so that's very interesting. In addition, PRDX3 is also regulated by MYC. So this is just going through the catalytic life cycle of the pararedoxin 3. So it neutralizes H2O2 to form the sulfonic acid intermediate state, and that in turn reduces the enzyme to disulfide bonds, and that gets turned back to its original state by other enzymes in the mitochondria. If there's too much H2O2, it becomes hyperoxidized, and it takes a slower time to return to its natural state because another enzyme has to be imported from the cytosol. So again, theostreptin is a cyclic oligopeptide antibiotic isolated from Streptomyces. Theostreptin is reported to dimerize those disulfide or those thiol residues, inactivating it. And I just show an example of that in the western below there. So a reasonable hypothesis would be interfering with PRDX3 activity would inhibit their ability to adapt to oxidative stress. So we predict that this will raise the mitochondrial ROS, as well as inhibiting proliferation in 2D and 3D, as well as in the PUMA assay. And then we eventually want to go into the in vivo setting. So this is just single agent activity in 3 cell lines, well, 2 cell lines and 1 PDX model from Alejandro's group. This is just a single agent activity. And to mimic the lung microenvironment, I add an exogenous inducer of oxidative stress called TBHP in this next assay. So we notice here in the presence of TBHP, tert-butyl hydroxyperoxide, when used in combination, the cells cannot tolerate that versus the single agent controls. Just to show that it does increase mitochondrial ROS, we did a mitosoc staining, and in all 3 models, we see that combination results in higher mitochondrial ROS versus single agent controls. And then here, I hope this time-lapse works. So this is looking at growth in vehicle under oxidative stress, under thiostreptin inhibition, and under combination. And as you can see that the cells can really tolerate this oxidative stressor, and they can tolerate thiostreptin, but when you add them in combination, that really reduces the proliferation of these cells. And then we see this in 3 cell models. We also wanted to do this in 3D because tumor cells act differently in 3D. Unique properties such as gradients of oxygen, CO2 nutrients occur in a 3D setting. So we do find that the combination results in increased tumor cell death. So the red is tumor cell death around those spheroids. And so the combination results in greater tumor cell death versus single agent controls. And then we decided to look at the PUMA assay, which is basically my go, no-go into assay before going into in vivo studies. So basically, we're studying how these tumor cells grow in viable lung tissue, and that's demonstrated here. So I learned this when I was at NIH with Chankana, and I learned it from Arnulfo Mendoza. So this is what it briefly looks like, just untreated. So we have tumor cells arriving day 0, and then over time you can see the evolution of the PUMA slice. So if we apply the drug, as we found that as low as 100 nanomolar was able to inhibit the growth of these tumor cells in the lung, ex vivo lung tissue, and we also see this in another cell line. So just a summary of the findings. So NRF2 in our previous study seemed to be very important in the metastasis biology, and NRF2 can actually influence a lot of aspects of mitochondrial biology. We find that if we inhibit PRDX3, which is downstream of NRF2, we find that it can recapitulate what we saw with an NRF2 knockdown. So ongoing studies include assessing the effects of thiostreptin in mouse models of metastatic osteosarcoma. And then we're also doing, since we want to do mechanism, we want to do knockdown. And of course, it's suggested by the clinicians that we collaborate with, we always want to do drug synergy studies with second-line chemotherapies like etoposide, ifosfamide, and gemcitabine. And with that, I'd like to thank you for your attention.
Good morning, everyone. Big thanks to MIB agents for this exciting opportunity for us to present our work here, and also for trusting us with their money and funding our research. Really appreciative of that. So our efforts are basically dedicated to honoring the memories of the osteoangels and the hopes that we will be able to help osteo warriors and osteoparents. So it's been known for some time that cancer cells alter their metabolism because of their excessive growth. And one of the ways they do it is by secreting glutamate and using glutamate to fuel their growth in an autocrine fashion. And for this, they use various different types of receptors. So our question was, if we simply inhibit the glutamate release and the signaling, so if you stop the glutamate release, block the signaling, then there won't be any growth at all. Sorry. So how do we block glutamate release? By using a drug called riluzole, Riluzole was FDA approved in 1995 for ALS, and it's available in various different forms. It's basically a benzothiazole drug. What does riluzole do? It blocks glutamate release. It also enhances the reuptake, so it clears the glutamate from the extracellular matrix, and also inhibits the activation of the receptors because there's no glutamate. Why glutamate? Because, as I said, it was FDA approved over 3 decades ago, so there's a lot of information about tolerance, toxicity, and pharmacokinetics, and also it's been shown to have promise in other cancers such as melanoma and glioma. When I started this work, this is the information we had, that the MG63 cells, which are osteoblast-like cells, were shown to release spontaneous glutamate release, and this glutamate release was blocked by riluzole. And little later than that, what was found was that in a European genome-wide association study of patient of European descent, that one of the glutamate receptors was highly expressed. So we went ahead to test this, and we published a paper in 2021, and prior to that, we also had a paper where we showed that riluzole blocks cell proliferation, induces apoptosis, and it does this by blocking the activity of the glutamate receptor, mGluR5. We actually looked into the mechanism of this, but I'm not going to go into the mechanism, but I'm going to just tell you the efficacy study. So we went further ahead because showing that the drug works in 2 cell lines isn't good enough, so we designed the study. Of course, this is done in collaboration with Dr. Healy, and we got this award together. Dr. Healy is the co-PI on the award. So we chose 7 established cell lines and 5 patient-derived models, which come from Dr. Healy's lab. As you can see there, we actually chose 3 sets of non-metastatic and metastatic models. The South 2LM7 cells are from Dr. Eugenie Kleinemer's lab from MD Anderson. The MG63 cells, 63.3 cells were provided by Dr. Oh my gosh, blanking off, from NCI, the one who got the award yesterday. LeBlanc, yes. Oh, thank you, yes, LeBlanc. Sorry. And the OS482 cells, which are mouse model of LM7, which were presented yesterday, they come from Dr. Stuart Orkin's lab. So besides these models, we also had a normal osteoblastic cell, which are basically immortalized fetal osteoblasts, used as our control. And then we have our PDX models listed below, which are 5 in number. So we wanted to first test the secretion of glutamate. As you can see, these are primary osteosarcoma cell lines. They all actually secrete glutamate in increasing amount over the period of 7 days. The metastatic ones, so we are comparing the levels to the fetal osteoblastic cell line as the control. So the levels at day 5 in primary are a little higher than for some of them, are higher than the fetal osteoblastic cell line. However, with metastatic cell lines, you see a significant increase in the amount of glutamate secretion, which tends to agree with the aggressive growth they show. All but 1 PDX cell line also showed glutamate secretion. Now, when we tested the efficacy of riluzole in blocking the cell growth in these cell lines, all of the cell lines and the PDXs showed growth in dose-dependent growth inhibition when we treated with riluzole. Now, when cells metastasize, they have to first invade the tissues and then migrate. And we basically use this assay called the Boyden chamber assay, which most of you may be familiar with, where the cells are seated in the upper chamber. In the lower chamber, they actually come to the lower chamber by crossing the barrier, which is shown in green. They cross it, it's a porous membrane. The cells, if they're able to migrate, they come to the other side, and then you can stain them. But if you need to test a drug, you can actually add the drug in the lower chamber. And if the drug blocks the activity, invasion activity, you're able to see the difference in the migration or the invasion ability. So we tested this. Nope, it went ahead. Yeah. So we tested this in most of the cell lines. You can see the OS cell lines on the left and the middle. The red bars are basically the control. The purple are the negative control. And you can see that compared to the red bars, you see a decrease in the invasion ability of all of the cells, including the PDXs, when you compare to the control samples. So this shows us that riluzole actually inhibits the invasive ability of these cells. Then we went ahead and tested prodrugs. Now, why prodrugs? Because when ALS is given, when Riluzole is given in ALS patients, it's given twice daily at 50 milligram dosage. And what happens is that because of the heterogeneity of one of the enzymes, which is in the liver, the CYP1A2, Riluzole gets metabolized and eliminated through the urinary tract system after hepatic clearance. And the interesting part is that the CYP1A2 enzyme is heterogeneous in the population, because of which the effective exposure of the drug after oral dosing is different in the human population. So pharma companies came up with this dipeptide drugs that are basically escape the hepatic clearance. And it's a scene that I've shown that within 2 hours, the both FC3423 compound and troriluzole get broken down into, converted to riluzole. And you can see on the right side that the FC3423 and troriluzole are effective in inhibiting the growth. This is in vitro work. Then we went ahead and we did in vivo work, where you see 4 groups of mice, control, riluzole, FC compound, and then troriluzole. And at the end, you can see at the 4 weeks, you can see that the tumors are markedly decreased. And this is basically an in vivo metastasis model, where we do tailvein injection for implanting the cells, which carry the luciferase gene. So that's why we can monitor the growth by bioluminescence. The bar graphs are shown on the right. We looked at the tissues for proliferative markers, and compared to the control, we see a significant reduction in the proliferation of riluzole, troriluzole, and the FC compound. And we then looked at the apoptotic activity by staining for activated caspase, and you can see that the activated caspase activity is elevated in the drug-treated samples, indicating that there's apoptosis. This slide is to show you that riluzole has been in clinical trials, either alone or with other agents, and basically to highlight the point that it works really well with other drugs. It's also been tested with so it's not just riluzole, but troriluzole also has been in clinical trials and has shown that it's very well tolerated. It's not a secret that monotherapies don't work, and we were not surprised to see that 3 of our cell lines showed a high IC50 value. I'm going to highlight just 2 of them, 143B and one of the PDXs. 143B contains basically a KRAS mutation, because of which the IC50 value is really high. So we basically use these cells and use a KRAS inhibitor, which is experimental one, the SI12-5. And at the bottom, you see the control cells, which is, they don't carry the KRAS mutation. They're not affected. However, in 143B cells, you see synergistic activity or inhibition of both drugs in inhibiting growth. So that was an experimental drug. Then we went ahead and tried a clinical drug, which is in clinical trials for pancreatic adenocarcinoma, the PDAC, and shown some promise in that cancer. So we used that inhibitor. I'm sorry, the title should read pan-KRAS inhibitor, which is the RMC6236. And we do see synergistic inhibition when we use in 143, but not in the control cell line. Now, the OS339, which didn't respond so well to riluzole, contains, basically has MCL1 amplification, which was talked about yesterday briefly. One of the awardees plans to work on this. So MCL1 blocks apoptosis, and because of which it's hard to kill the cells. But if you use an inhibitor, you can actually promote apoptosis. So we basically combine riluzole with MCL1 inhibitor, and we see a synergistic effect. This is just one set of data, but the other ones are complete, so we just need to do more of these. So basically, to summarize, riluzole shows block in proliferation as well as induces apoptosis. The prodrugs work really well, and both in vitro and in vivo, and it seemed to work really well with combination with other drugs. And I want to thank my students. Oken, Mariam, and Shraddha was my tech, and they're the one who have done most of this work. And of course, big thanks to Dr. Healy for trusting us with these PDX samples and his contribution to the project. And Raj is the one who has actually made the PDXs. So, and I hope that you saw Pooja's poster yesterday. It's still on. If you have time, please visit her, ask her questions, or ask me questions, and we'd be happy to discuss further. And big thank you to all for being here. Thank you.
Good morning, everybody. Thank you so much for the opportunity to speak to you all today. My name is Sasha Blay. I'm a senior PhD student in the Sloan lab at the Hospital for Sick Children, Toronto, Canada. Okay, I don't need to reiterate why we're here today, but I do want to emphasize that we have not made major advances in the clinical treatment of this disease since the 1980s, when we added cisplatin, an alkylating agent, to the clinical regimen. And of course, this is underscored by complete genomic chaos and complexity, making it difficult for us to understand the biology, and additionally then to extrapolate how we can better treat, prognosticate, etc. My understanding of the literature is that there's a couple different camps when it comes to the evolution of osteosarcoma. There's data that supports a model of branching and parallel evolution, where you start out with a lot of heterogeneity that is gently enriched or diminished different subclones over time, in contrast to a more punctuated model of evolution, where you have quiet periods that are suddenly interrupted by large, catastrophic, chromoplectic, etc. events. And so I want to understand the evolution, the whole picture of osteosarcoma progression from very early tumor genesis events, understanding how the mutational landscape changes under chemotherapeutic pressures, and then finally the outgrowth of these post-treatment populations. So I invite you to join me on what I'm calling a pathway to chemoresistance. Now, of course, this is only possible with as large a cohort as I possibly could. And so I curated this cohort comprised of 76 pediatric patients. This is 76 patients, 100 whole genome samples, all of which have a matched normal. And of course, this was only possible with the help from friends across the Sick Kids Institute and around the world. It was very important that my cohort reflect advanced disease. I need to make sure that I have samples that are both primary, initial, or untreated, as well as the more non-primary advanced cases that have been treated with either MAP or some sort of variant. Now, just from a high-level bioinformatic overview, my findings recapitulate a lot of what we already see in the literature. We have really high numbers of copy number losses, which end up disproportionately hitting our tumor suppressor genes and these major players. Surprisingly, the tumor mutation burden was a little higher than I expected. Almost 1/3 of all of the tumor samples were categorized as either hyper or ultra hypermutant, carrying more than 10 mutations per megabase, which is more than you might expect. And of course, this was skewed towards the non-primary samples. Additionally, whole genome duplication was surprisingly pervasive in almost 60% of my samples. And when I actually scored each autosome on its own as whether it was duplicated or not, I find that there was a very nice bimodal distribution here. Samples were either very much not duplicated or very much duplicated. And again, this was skewed towards the non-primary samples. So in order to understand the development of chemoresistance, I've harnessed mutational signatures. These are patterns of somatic mutations in the cancer genome attributed to specific endogenous or exogenous etiologies. So we see UV signatures in melanoma, tobacco signatures in lung cancer, but we also have mutational signatures for a number of different mutagenic chemotherapies, like cisplatin. The way that this alkylating agent works is creating very specific adducts in the DNA that upon replication and the cells trying to repair this damage leads to specific types of point mutations. And when we sequence the genome, we can see this mutational spectrum here, very reminiscent of the drug's original mechanism of action. So this is SPS31, one of our platinum-associated signatures. And for me, if a cell has been exposed to the chemotherapy, it's survived and it's proliferated up to the detection of our whole genome sequencing, to me, this cell must be chemoresistant. It's carrying the battle scar of its exposure to the platinum chemotherapy. So our lab has worked extensively to characterize all of the therapy-associated mutational signatures, pan-pediatric cancer, but I want to know specifically what this looks like in osteosarcoma. And it's also a great model because it's so regimented that most of the patients receive cisplatin or some variant of it. And so starting out with the platinum-associated signatures, I found that a total of 28 of my samples, I was able to detect the signature. This, of course, was much more prevalent in the non-primary, so your metastasis and relapse later samples. And in a subset of 33 samples that I was for sure knew very nicely clinically annotated exposed, about half of them developed the signature. And we must remember that these alkylating agents can contribute a lot of damage, and average more than 3,000 SNVs in these samples, and accounting for up to a third of the total mutational burden. You can see that in red, the platinum-associated signatures were one of the top active mutational processes in these tumors. So you can imagine the implications for secondary treatment-associated malignancies, actually inducing additional drivers, et cetera. So in order to understand how the therapy is, in order to understand how the clonal evolution is changing in the context of therapy, I devised this pipeline. Normally, we extract mutational signatures at the bulk level, all of the variants in the sample, but what I've done is first subsetted all of the variants into different subclones, and it's there at the subclonal level that I extract mutational signatures. So when I reconstruct the phylogenetic trees, I can elucidate how the mutational processes are changing over space and time. And so, sure enough, here are some example trees where I do detect a signature in the samples. And in red here are the subclones in which I detect a platinum-associated signature. I can pinpoint very specifically where on the tree the cells begin to feel the effects of therapy. And you'll notice that this is specific to either one or two of the clonal branches and appears to be fairly subclonal late towards the terminal ends of the branches. Now that I can pinpoint very specifically which subclones are feeling the effects of therapy, I can begin to ask specific questions about the timing of events. So let's take one example here where this patient had a primary in the leg that metastasized twice to the lungs and additionally to the mediastinum. And I propose two different simplified scenarios. The first is which you would feel the effects of therapy relatively early in the primary prior to an evolutionary split, not a physical dissemination, but just a splitting off into each of the metastatic clones. And in this scenario, the signature should be shared between all of the sites. Alternatively, if you were to evolutionary split from the primary preceding the effects of therapy, then each of the metastases might have their own signatures comprised of unique sets of mutations. And so this second scenario is exactly what I see in 11 patients with multiple samples. This is the same example as before, and my red subpopulations there are all completely unique in all the 3 different metastatic sites. This seems to be true regardless of where the metastatic sites in the body. And so what I'm seeing is that all of the metastases are independently developing the mutational signatures. And so this evolutionary split must have preceded this effect. Additionally, I tracked these clones between time points, and I found that the clones that did have a platinum-associated signature were becoming enriched. They were increasing in prevalence in cancer cell fraction over time, in contrast to the clones that did not have a signature. And in my last minute, I want to tell you some very exciting results from a little pilot experiment of mine, harnessing some banked plasma samples and sequencing ctDNA. I want to ask more questions about the timing and the events using the mutational signatures as a proxy for chemoresistance status. So my first question, kind of a positive control, is can we detect the platinum signature robustly in the plasma? Does it look like what we see in the bulk tumor? And additionally, what more can we learn about the clonal evolution from the ctDNA, which actually might be capturing more cells from all different parts of the tumor as it sheds? And so I have just, I got the fast cues back just a couple weeks ago. This is hot off the press. At the top here, you'll see a mutational spectrum of over 10,000 shared mutations found in both the bulk and the plasma that looks exactly like my reference platinum signature SBS 31 on the bottom, which proves that I'm getting exactly the same mutational signatures, in fact, the very same mutations in both the bulk and the plasma. Additionally, I noticed that when I reconstructed the evolutionary trees, we have the shared mutations, but I found more than 5,000 SNVs that were specific just to the ctDNA. And so we're capturing additional heterogeneity that you might miss just by a single bulk sample. This might include additional information, targetable new drivers. And it actually, from this sample, stems from one specific lineage. So we're learning a little bit more about the tumor's evolution as well by incorporating ctDNA into the bulk analysis. And so I'm excited now with this positive control to apply this to serial samples so that I can pinpoint exactly when the signature emerges and as well how long it's present in the body and the blood. Okay. Before I get flag shamed, I just want to summarize that for the scenario of the metastasis feeling the effects of the therapy, this is a very interesting example of convergent evolution to a chemoresistant phenotype. And it makes me wonder if there's some inherent survival advantage possessed by these metastatic lineages determined very early in tumor evolution that is simply selected for by the chemotherapeutic bottleneck. Dare I say these are the anchor cells that we heard about yesterday, and that platinum contributes substantially to the outgrowth of post-exposed populations, and that plasma can be very informative in addition, or perhaps even as a proxy for determining chemosensitive status without invasive testing. Thank you so much. I look forward to your questions.
Thank you so much for having me today. My name is Chelsea Burke, and I'm a pediatric oncologist at Stanford University, and I'm going to be talking today about tumor evolution and mechanisms of resistance in osteosarcoma. So as my fellow panelists have touched upon, with introduction of MAP chemotherapy in the 70s and 80s, we did see significant improvements in survival for patients with localized osteosarcoma. But as we know, progress has largely plateaued over the last several decades, and outcomes remain poor for those that have metastatic or relapsed disease. And so resistance to our standard treatments has become a major barrier to effective care, which is likely driven in part by osteosarcoma's underlying genomic complexity. And so we know the genome of osteosarcoma is chaotic and unstable. This leads to complex biology and variable treatment responses. And so we also know that osteosarcoma has very few actionable or targetable mutations, and so identifying these novel therapeutic strategies is a major focus in improving outcomes. MAP chemotherapy still remains our standard frontline treatment for osteosarcoma, but we really lack a full understanding of how these drugs affect tumor biology at the cellular level. So what is actually happening to the tumor cells when we give MAP chemotherapy? Why do some tumors respond while others become resistant and go on to metastasize? And so one prevailing theory is that chemotherapy imposes a form of selective pressure, which eliminates these drug-sensitive cells but allows resistant populations to go on and survive and expand. And over time, it's these resistant cell populations that become the dominant populations that drive relapse and disease progression. And so this image at the bottom is from work done by Dr. Michael Kinnaman that was published a couple of years ago, where he took osteosarcoma patient samples and saw that there were these small populations of resistant cells that emerged following treatment. and dominate at recurrent sites. And so this work kind of underscores a shift in how we think about treatment failure in osteosarcoma, not simply as tumor eradication or incomplete tumor eradication, but as a biological process in which resistant cells are actively selected for and expand over time. So with advances in things like single-cell sequencing analysis over the last several years, we're able now to identify and better characterize these small populations. And we're understanding cancer not just as kind of a collection of rapidly replicating cells, but as a complex system made up of distinct transcriptionally defined cell states, each having their own unique kind of properties and vulnerabilities. And so Dr. Jovana Pavicic has done a lot of work in single-cell sequencing, trying to define and characterize these distinct cell states and see in osteosarcoma how they respond to treatment. And so once we're able to kind of characterize these cell states, how then can we effectively target these resistant cells? Again, work by Dr. Pavicic and other colleagues at Columbia University have shown that these cancer cells rely on complex regulatory networks in order to maintain their malignant cell state. And these states are maintained actually by a relatively small number of key proteins, or what are termed master regulators. And these master regulators act at critical control nodes or bottlenecks that are essential for preserving the cancer cell's identity and survival. And so once we can identify and therapeutically target these master regulators, we kind of disrupt the entire cell architecture, hopefully inducing cell death. And so by applying a computational approach called VIPER, we're able to kind of identify these top rank master regulators for each of these cell states and identify drugs that could be used to target them. And so all of this has kind of laid the groundwork for our project, where we're really trying to look at the effect of MAP chemotherapy on a cellular level in osteosarcoma tumors. Hopefully by identifying these small chemotherapy-resistant populations, we can understand them better and then target them. And so in order to study MAP chemotherapy, we use our PDX models, our patient-derived xenograft models. We take diagnostic tissue from our osteosarcoma patients. These are ones at MSK, and implant them into our mice and give them MAP chemotherapy. And so that is really the first hurdle of this project. And in order to kind of model treatment accurately, it was important to replicate clinically relevant MAP chemotherapy dosing in our mouse models. And so help from our colleagues at St. Jude had previously established some human equivalent dosing in mice, which I show here. Once we were able to kind of confirm tolerability, we were able to administer a full cycle of MAP chemotherapy in our PDX models. Very similar to what we have to do in our patients. They also require a lot of supportive care, so extra fluids, leucovorin, that kind of thing. And so when treating, or after treating these PDX models, we collected tumor tissue at 2 time points. So the first was at week 3 following after the mice had received 2 days of cisplatin and doxorubicin, and then at a second time point when the PDXs had received the full cycle, so including high-dose methotrexate. And up to this point, we've treated 2 of our distinct PDX models, but I have some information for one of those models here. So this is one of the tumor volume graphs of the PDXs that we treated, also showing kind of the timing of when we did our tissue harvest here. And we did, see some initial tumor regression after giving the cisplatin and the doxorubicin, but as you can see, kind of towards the end of week 3, before we give the high-dose methotrexate, we do notice that there is some tumor regrowth, which may be reflective of an underlying resistant population that is emerging. And so this is really just some preliminary data that we've just getting back in the last week or so. But we have gene expression analysis that's profiling more than 260,000 single nuclei across 4 samples that we collected from one PDX model. So the UMAP here shows unsupervised clustering of the nuclei by gene expression. And these are some of our preliminary cell annotations that you can see here for our kind of different cell clusters. And so in the blue, we have our proliferative cells, which are cells that are replicating and in the cell cycle. In the red, we see this cluster that has expression of extracellular remodeling genes and genes that are involved in osteoblast differentiation. In the green, we see expressions of genes that are allowing survival in hypoxic conditions and show markers for chondrocyte differentiation. And then finally, in purple, we see this cluster, which really represents like stressed or apoptotic state of genes that are involved in cell death. And so in the bar plot on the right, this shows the proportion of cells belonging to each of those clusters, and we can see how this really varies between different replicates for each treatment arm. So kind of just highlighting the importance to sample multiple replicates even within the same PDX model. And so taking the data from the next slide, we're then able to color these cells by treatment condition. So we see our vehicle mice, those that have received cisplatin and doxorubicin, and those that have gotten a full cycle of MAP chemotherapy. And then again, the bar plot on the right showing the mean proportion of the cells belonging to each treatment condition in each of these clusters. And again, some of this is preliminary data, but it seems to suggest that there is a decrease in that cluster 0, which was that ECM active mesenchymal or osteoblast prime state. We seem to show a relative increase in the hypoxia adapted chondroblast-like state. And specifically in the cisplatin doxorubicin, we see that the stressed or apoptotic cell state is particularly enriched there, which is, I think what we would expect after giving agents like cisplatin and doxorubicin. And I know I am running a little bit short on time, but also just want to touch on kind of now that we're able to understand these cell states a little bit more, we really want to look at the regulatory drivers of each of these cell states. So this is just kind of an example of further directions of this project where we will use that Viper algorithm to identify, again, those master regulators of those enriched cell states, and hopefully then use agents that are either FDA approved or that can be repurposed to target these master regulators. So these are the ongoing directions. Again, this is a lot of new data that we are still analyzing, so I'll be excited to kind of share more about this in the future. But I want to make sure to recognize everyone that's been involved in this. So none of this could have been done without Ava and the Levin family. Thank you to all of the osteo warriors and the osteo angels here. And I have to say thank you so much to the Dela Cruz Lab at MSK, who has continued to do this work even though I have transitioned to the West Coast now, and Dr. Jovana Pavlicic, who's been integral in our single cell analysis. So thank you guys so much for having me today.
Thank you very much. I see quite a few questions on Slido. Why don't we have about 8 minutes for questions. So let's take that top one, Dr. Scotlandi, and then Dr. Lizardo, kind of move down. All right, go ahead. first question is, Dr. Roberts asking about the discordance between the mice and human studies, mentioning that trabectedin, it was found that a different schedule and dosing overcame that difference with added information. Is there a...
I mean, I think that the major reason of discordance is that the patients that were treated in the phase 2 clinical study are really heavily pretreated patients. No? So it's very difficult to see an efficacy of a drug when the condition is so advanced. So, and the second reason, in my opinion, is that, yeah, we know that ixabepilone is a tubulin stabilizer, but we don't know actually what does it mean inside in terms of signaling, intracellular signaling. So for me, it was intriguing to see such a strong inhibition in this cell line with this huge amplification of MYC, as I said. So what I would like to explore is if these microtubule can actually change the shuttle of MYC from the cytoplasm to the nucleus, so they can change the MYC activity in terms of a transcriptional factor. And so the second part of the question is if we can redesign. Yeah, other example of redesign was Ewing intrabectul. I think yes, we can try to pursue in the same way for ixabepilone.
Go ahead, Josh.
Yeah, Sasha, great talk this morning. Can you share with us a bit your thoughts on what you touched on the end about that unique evolutionary branch from the ctDNA? Does that, do you think, represent a new metastasis that wasn't detected, or is that from within the primary tumor that just wasn't seen on the original sequencing?
Thank you so much. Absolutely, it's very difficult to say. Because of the clonal evolution, I'm able to tell which lineages, which progenitors have led to the ctDNA, and I think it's interesting that these are relegated just from what I can see to one specific lineage, and I see this in other pediatric cancers as well. It's usually just one lineage that I can connect to the ctDNA. I'm limited by what we can do with whole genome sequencing, and speaking to some of these Slido questions as well, I think we might have to go either into single cell or using barcoding techniques to try and understand exactly which cells are leaving, which cells are sticking to the primary tumor, because in theory, these are the lineages that are going on to see the metastases, right? These are the ones that are disseminating in the blood, and I can see that these are part of the active tumor. I don't believe that these are the dead cells. I believe that these were previously living, replicating parts of the tumor, and so I'm absolutely looking to validate this effect. Thank you.
Let's do Dr. Lizardo.
So for Ryan Roberts' question regarding is this the best approach to study metastasis regarding studying human tumor cells in small mouse lungs. So I would say we would have to make our experimental approach as versatile as metastatic tumor cells are. So for example, I would gladly collaborate with Heinrich Kovar in Europe. So what he does is he co-cultures tumor cells with lung organoids. So I would be happy to incorporate that model in my studies as well. So I think the mouse lung does represent like a general mammalian, structure of lungs, but I think it'd also be informative to have human microvascular endothelial cells and alveolar macrophages to be co-cultured with your metastatic tumor cells and see how they. Okay. Yeah. And then see how they do with and without your new therapy. So.
We have a microphone over here. Go ahead, Dr. Kleinerman.
Thank you. My question is for Dr. Mahajan. Really elegant, intuitive work. So I'm wondering if there's a lot of thought right now that the tumor acidic and hypoxic microenvironment actually is the thing that paralyzes T cells and inhibits immunotherapy. Have you had any thought about using your drug and seeing if it can augment the effects of immunotherapy and actually reverse the suppressive effect of the tumor microenvironment?
Thank you very much. We haven't looked at directly, but there has been one paper recently where riluzole has seemed to have some immunomodulatory effect, especially acting on the G-gasting pathway. So I think that's promising, but we haven't looked into that directly ourselves, but something to pursue in the future. Thank you.
Jeff?
Sasha, I have another question for you. Sorry.
Where are you? Oh,
over
okay.
here. Really interesting talk, and I know it made sense to focus on the cisplatin signature clades from your trees, but do you have a sense of what's going on in those others that didn't bear the cisplatin signature? Because they've obviously also survived cisplatin, and what's helping them survive?
Yes, this is a question that I get a lot because there are absolutely populations that seem to just be hanging out. I detect them in the primary, and then I detect them in the non-primaries as well. And so I see this disparity on the subclonal level, but we also see it on the bulk tumor level. Why do only some of the samples develop the signature despite the stars aligning, like sufficient exposure, sufficient cycles, sufficient dosage? And so that's very much a big question that we have, and I might attribute that to different mechanisms of drug resistance. You know, perhaps there's different populations that the drug doesn't actually touch or ever get into the nucleus. Maybe it's being effluxed or something. Maybe it's a little hidden tight niche right in the center of the tumor. I don't know, but it's a very important question for why are these populations surviving? Why are these diminished, and why do these survive? And I've tried to elucidate patterns across different samples and across different subclones, and I really haven't found anything that stands out. So absolutely still a large remaining question.
So they don't have like an anthracycline signature? It's not like they were hit by something else?
No, sometimes I see the 5FU signature, but this is even less prevalent than the cisplatin. So.
Rosie, and then I'll ask a final question.
Yeah, this is a question for Chelsea. I really enjoyed the talk. It's more about you mentioned the ECM remodeling type. It's interesting that you see it. That you don't see it in the if I understood it correctly, in the C1, the post-treatment compared to the baseline. And I think, at least when the data that I have looking at like metastatic osteo, they're very matrix remodeling and active. But in a way, you'd think if they were more mature, maybe they wouldn't be doing that. So I just wondered your thoughts on that.
Yeah, I still think we're trying to kind of elucidate that, and I think a big part of our project will also be to look at these cell states in patient samples. Again, this is from the diagnostic sample, but we also have kind of longitudinal samples at their definitive resection and at their places of metastatic relapse. So I think as we kind of gather more information from those, we'll get a better idea. But I think we still need to explore that a little bit. Thanks.
Fantastic. That's perfect. We're going to wrap up this session. We are out of time. I'm getting flagged. Thank you.